Hi everybody, in this video we're going to talk about bacterial cell walls, and then we're going to get into some of those internal structures if we have time. Okay, so the cell wall is a strong substance that protects cells from lysosing open. So lysis is breaking open. The cell wall basically helps them so that they don't break open. Bacteria have two distinguishable types of cell walls. And this breaks them into two groups. Either they are considered gram-positive or they are considered gram-negative. The difference is associated with the amount of peptidoglycan in the cell wall. Gram-positive bacteria have thick, multiple layers of peptidoglycan, whereas gram-negative bacteria have very thin, only a few layers of peptidoglycan, and you can see it out here, okay? That's not the only difference in their cell, but a gram stain differentiates these two based on the peptidoglycan layers. So peptidoglycan then is a carbohydrate protein structure that is layered in your bacteria. Bacteria can have as many as 30 layers of peptidoglycan, also known as PTG, just so you know. Within the PTG, there is teichoic acid. So gram-positive bacteria have teichoic acid built in or found within the peptidoglycan layers. Peptidoglycan actually causes cells to be more prone to certain types of antibiotic damage. So peptidoglycan is the target for penicillin as well as for lysozyme. And because of that, if your cells have a lot of peptidoglycan, then you're going to be very sensitive to these two chemicals. And you're going to more than likely not be able to produce the cell wall properly, and then you will die. So what does penicillin do? Penicillin binds to the proteins that build the peptidoglycan cell wall. And because of that, this is going to be much more effective against gram-positive bacteria because gram-positive bacteria have multiple layers, up to 30 layers of peptidoglycan. So, and that's where this cute little cartoon comes into play, the bold and the bacterial. Here's some penicillin, or here's a bacterial cell, here's penicillin, and they love each other. There's mom and dad freaking out because, well, he's penicillin, they can't be in love. Yeah, haha, kind of funny. Anyways, penicillin derivatives have been produced to work against gram-negative cells because gram-positive, or because typical penicillin doesn't affect gram-negative cells, since there's very few layers of peptidoglycan, and that's what penicillin targets. And so how does it actually work? This is for both penicillin and lysozyme, okay? So lysozyme breaks bonds within the peptidoglycan layers, and so the peptidoglycan is no longer stable. Penicillin inhibits the production of those layers, and so there's no cell wall produced. And so what happens then, and that's what I just said here. So what happens then, there we go, the cells become very sensitive to the solution in the environment. So this is known as a protoplast. It no longer has that cell wall surrounding it to protect it, okay? And this cell can survive in an isotonic environment very easily, but if the environment becomes hypertonic or hypotonic, problems occur. So in this case, the cell in a hypertonic solution would crenate, just shrivel up. In a hypotonic solution, that's where the real big problems happen. water is going to flow into the solution, into the cell swells up, but without that cell wall, the cell is going to burst open and die. So we have these thick peptidoglycan layers, multiple layers of peptidoglycan with teichoic acid built in your gram-positive cells. Gram-negative cells have only 1 or 2 layers of peptidoglycan. That's it. So their cell wall is not protected by just the peptidoglycan. They actually have an extra plasma membrane. This is called the outer plasma membrane. So they have the normal plasma membrane, which all cells have, and then they have this outer plasma membrane, which is part of their cell wall. This outer plasma membrane contains a lipopolysaccharide within it, and that lipopolysaccharide has multiple parts. So you have an O antigen, you have a lipid A. Those are the two really important parts, okay? And then in between, I did that. Yes, I did. Sorry. I didn't mention this. In between the inner membrane and the outer membrane, this area is called the periplasmic space, okay? So the outer membrane has that lipopolysaccharide, and this is important medically because it actually, the lipopolysaccharide has an endotoxin known as lipid A. Lipid A is an endotoxin, and when it's released, it can cause your body temperature to rise to very high temperatures that would cause you to go into shock. It also has that O protein chain, and that O side chain is able to help you identify different strains of the same species. And so the lipopolysaccharide, whereas teichoic acid was what gave your gram-positive bacteria the negative charge, the lipopolysaccharide gives the bacteria their negative charge. We have all these proteins with these lipopolysaccharides in them. The O polysaccharide side chain. This is what we use to help identify different strains of bacteria. So we have good E. coli. Everybody has E. coli in their gut, and the E. coli in their gut actually protects them and helps them to stay healthy and produce certain vitamins whereas there's also pathogenic strains of E. coli that have a toxin known as Shiga toxin, and Shiga toxin causes hemorrhagic colitis, where your colon will start to bleed. So it's really dangerous. E. coli 0157H7 is the pathogenic strain of E. coli. That's not the type of E. coli that we have in our gut normally. This is what we get when we eat food that has been undercooked, that has been, I don't know, where the animal was sick, or where the food was cooked improperly, whatever. Okay. Then we have lipid A. Lipid A is a portion of the lipopolysaccharide that helps to anchor the molecule. It is an endotoxin, and it's released by all gram-negative bacteria, and it's released when the bacteria are killed. And so the more bacteria that are gram-negative in your system, the more of these lipid A molecules that are going to be released, these endotoxins. And each endotoxin then enters your blood and moves to your hypothalamus. The hypothalamus is the region of the body that controls temperature. And so every single one of these lipid A endotoxins stimulates the hypothalamus to increase the temperature. And if you increase your temperature too much, this can lead to endotoxic shock, which will then kill you. Well, I won't say will, can kill you. So not everything does kill, but a lot of things can kill you. So you can die in a lot of different ways. Scary, right? So there are some bacteria that lack a cell wall. So I told you that in general, we assume all bacteria have a cell wall, but just like everything, there's always the exception to the rule. And so in this case, mycoplasma are our exception. Mycoplasma are lack the cell wall. They're considered gram-positive bacteria, but because they lack the cell wall, they don't stain gram-positive, if that makes sense. They don't have the peptidoglycan, so they would stain and look pink, but they're still put into the gram-positive grouping for other reasons. Because they have no cell wall, penicillin and lysozyme have no effect on them. The cytoplasmic membrane contains sterols that increase strength. So just like we have sterols in our cells, bacteria have their own sterols. And these sterols help to maintain the strength and rigidity of the plasma membrane. Because they have no cell wall, they also have multiple shapes. So they are called pleomorphic. They're able to change in shape. So you see them, here's your mycoplasma. They might be circular, they might be rod-shaped, they might form a structure that looks very similar to maybe a protozoan or other organism. Mycoplasma pneumonia, which is one species that we typically talk about in this class, is the causative agent to walking pneumonia, which is a milder form of pneumonia, but it's still a disease that we can get. So when we look at the Archaeans, they have different structures in their cell wall. So they don't have the peptidoglycan, but some do have a similar molecule, so similar in structure, but not exactly the same, which is known as either pseudopeptidoglycan or pseudomurine. But not all Archaeans have pseudomurine in their cell wall. Their cell walls tend to have other structures that allow them to survive in certain extreme environments, like extra salt transporters or temperature mechanisms within the cell wall that allow them to survive in more extreme environments. Okay, I'm going to stop here because we've talked about the cell wall and it's been 13 minutes. So I'm going to stop the video and then we'll get into some other structures of the cell in our next video. Bye.